The origin of COVID: did people or nature open Pandora's box at Wuhan? Bulletin of the Atomic Scientists. By Nicholas Wade. Published May 5, 2021. Narrative by Skeptical Waves. The COVID-19 pandemic has disrupted lives the world over for more than a year. Its death toll will soon reach 3 million people. Yet the origin of pandemic remains uncertain. The political agendas of governments and scientists have generated thick clouds of obfuscation, which the mainstream press seems helpless to dispel. In what follows, I will sort through the available scientific facts, which hold many clues as to what happened, and provide readers with the evidence to make their own judgments. I will then try to assess the complex issue of blame, which starts with, but extends far beyond, the government of China. By the end of this article, you may have learned a lot about the molecular biology of viruses. I will try to keep this process as painless as possible. But the science cannot be avoided because for now, and probably for a long time hence, it offers the only sure thread through the maze. The virus that caused the pandemic is known officially as SARS-CoV-2, but can be called SARS-2 for short. As many people know, there are two main theories about its origin. One is that it jumped naturally from wildlife to people. The other is that the virus was under study in a lab, from which it escaped. It matters a great deal which is the case if we hope to prevent a second such occurrence. I'll describe the two theories, explain why each is plausible, and then ask which provides the better explanation of the available facts. It's important to note that so far there is no direct evidence for either theory. Each depends on a set of reasonable conjectures but so far lacks proof. So I have only clues, not conclusions, to offer. But those clues point in a specific direction. And having inferred that direction, I'm going to delineate some of the strands in this tangled skein of disaster. A tale of two theories. After the pandemic first broke out in December 2019, Chinese authorities reported that many cases had occurred in the wet market, a place selling wild animals for meat in Wuhan. This reminded experts of the SARS-1 epidemic of 2002, in which a bat virus had spread first to civets, an animal sold in wet markets, and from civets to people. A similar bat virus caused a second epidemic, known as MERS, in 2012. This time the intermediary host animal was camels. The decoding of the virus's genome showed it belonged to a viral family known as beta coronaviruses, to which the SARS-1 and MERS viruses also belong. The relationship supported the idea that, like them, it was a natural virus that had managed to jump from bats, via another animal host, to people. The wet market connection, the major point of similarity with the SARS-1 and MERS epidemics, was soon broken. Chinese researchers found earlier cases in Wuhan with no link to the wet market. But that seemed not to matter when so much further evidence in support of natural emergence was expected shortly. Wuhan, however, is home of the Wuhan Institute of Virology, a leading world center for research on coronaviruses. So the possibility that the SARS-2 virus had escaped from the lab could not be ruled out. Two reasonable scenarios of origin were on the table. From early on, public and media perceptions were shaped in favor of the natural emergence scenario by strong statements from two scientific groups. These statements were not at first examined as critically as they should have been. We stand together to strongly condemn conspiracy theories suggesting that COVID-19 does not have a natural origin, a group of virologists and others wrote in The Lancet on February 19, 2020, when it was really far too soon for anyone to be sure what had happened. Scientists overwhelmingly conclude that this coronavirus originated in wildlife, they said, with a stirring rallying call for readers to stand with Chinese colleagues on the front line of fighting the disease. Contrary to the letter writer's assertion, the idea that the virus might have escaped from a lab invoked accident, not conspiracy. It surely needed to be explored, not rejected out of hand. A defining mark of good scientists is that they go to great pains to distinguish between what they know and what they don't know. By this criterion, the signatories of the Lancet letter were behaving as poor scientists. They were assuring the public of facts they could not know for sure were true. It later turned out that the Lancet letter had been organized and drafted by Peter Daszak, president of the Eco-Health Alliance of New York. Daszak's organization funded coronavirus research at the Wuhan Institute of Virology. If the SARS-2 virus had indeed escaped from research he funded, Daszak would be potentially culpable. This acute conflict of interest was not declared to the Lancet's readers. To the contrary, the letter concluded, "We declare no competing interests." Virologists like Daszak had much at stake in the assigning of blame for the pandemic. For 20 years, mostly beneath the public's attention, they had been playing a dangerous game. In their laboratories they routinely created viruses more dangerous than those that exist in nature. They argued that they could do so safely, and that by getting ahead of nature they could predict and prevent natural spillovers, the crossover of viruses from an animal host to people. If SARS-2 had indeed escaped from such a laboratory experiment, a savage blowback could be expected, and the storm of public indignation would affect virologists everywhere, not just in China. It would shatter the scientific edifice top to bottom, an MIT Technology Review editor, Antonio Regalado, said in March 2020. A second statement that had enormous influence in shaping public attitudes was a letter, in other words an opinion piece, not a scientific article, published on March 17, 2020 in the journal Nature Medicine. Its authors were a group of virologists led by Christian G. Anderson of the Scripps Research Institute. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or purposefully manipulated virus, the 5 virologists declared in the second paragraph of their letter. Unfortunately, this was another case of poor science, in the sense defined above. True, some older methods of cutting and pasting viral genomes retain telltale signs of manipulation. But newer methods, called no-seum or seamless approaches, leave no defining marks. Nor do other methods for manipulating viruses such as serial passage, the repeated transfer of viruses from one culture of cells to another. If a virus has been manipulated, whether with a seamless method or by serial passage, there is no way of knowing that this is the case. Anderson and his colleagues were assuring their readers of something they could not know. The discussion part of their letter begins, "It is improbable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-like coronavirus." But wait, didn't the lead say the virus had clearly not been manipulated? The author's degree of certainty seemed to slip several notches when it came to laying out their reasoning. The reason for the slippage is clear once the technical language has been penetrated. The 2 reasons the authors give for supposing manipulation to be improbable are decidedly inconclusive. First, they say that the spike protein of SARS-2 binds very well to its target, the human ACE2 receptor, but does so in a different way from that which physical calculations suggest would be the best fit. Therefore, the virus must have arisen by natural selection, not manipulation. If this argument seems hard to grasp, it's because it's so strained. The author's basic assumption, not spelt out, is that anyone trying to make a bat virus bind to human cells could do so in only one way. First, they would calculate the strongest possible fit between the human ACE2 receptor and the spike protein with which the virus latches onto it. They would then design the spike protein accordingly, by selecting the right string of amino acid units that compose it. Since the SARS-2 spike protein is not of this calculated best design, the Anderson paper says, therefore it can't have been manipulated. But this ignores the way that virologists do in fact get spike proteins to bind to chosen targets, which is not by calculation but by splicing in spike protein genes from other viruses or by serial passage. With serial passage, each time the virus's progeny are transferred to new cell cultures or animals, the more successful are selected until one emerges that makes a really tight bind to human cells. Natural selection has done all the heavy lifting. The Anderson paper's speculation about designing a viral spike protein through calculation has no bearing on whether or not the virus was manipulated by one of the other 2 methods. The author's second argument against manipulation is even more contrived. Although most living things use DNA as their hereditary material, a number of viruses use RNA, DNA's close chemical cousin. But RNA is difficult to manipulate, so researchers working on coronaviruses, which are RNA-based, will first convert the RNA genome to DNA. They manipulate the DNA version, whether by adding or altering genes, and then arrange for the manipulated DNA genome to be converted back into infectious RNA. Only a certain number of these DNA backbones have been described in the scientific literature. Anyone manipulating the SARS-2 virus would probably have used one of these known backbones, the Anderson group writes, and since SARS-2 is not derived from any of them, therefore it was not manipulated. But the argument is conspicuously inconclusive. DNA backbones are quite easy to make, so it's obviously possible that SARS-2 was manipulated using an unpublished DNA backbone. And that's it. These are the 2 arguments made by the Anderson group in support of their declaration that the SARS-2 virus was clearly not manipulated. And this conclusion, grounded in nothing but 2 inconclusive speculations, convinced the world's press that SARS-2 could not have escaped from a lab. A technical critique of the Anderson letter takes it down in harsher words. Science is supposedly a self-correcting community of experts who constantly check each other's work. So why didn't other virologists point out that the Anderson group's argument was full of absurdly large holes? Perhaps because in today's universities speech can be very costly. Careers can be destroyed for stepping out of line. Any virologist who challenges the community's declared view risks having his next grant application turned down by the panel of fellow virologists that advises the government grant distribution agency. The Daszak and Anderson letters were really political, not scientific, statements, yet were amazingly effective. Articles in the mainstream press repeatedly stated that a consensus of experts had ruled lab escape out of the question or extremely unlikely. Their authors relied for the most part on the Daszak and Anderson letters, failing to understand the yawning gaps in their arguments. Mainstream newspapers all have science journalists on their staff, as do the major networks, and these specialist reporters are supposed to be able to question scientists and check their assertions. But the Daszak and Anderson assertions went largely unchallenged. Doubts about natural emergence. Natural emergence was the media's preferred theory until around February 2021 and the visit by a World Health Organization, WHO, commission to China. The commission's composition and access were heavily controlled by the Chinese authorities. Its members, who included the ubiquitous Daszak, kept asserting before, during, and after their visit that lab escape was extremely unlikely. But this was not quite the propaganda victory the Chinese authorities may have been hoping for. What became clear was that the Chinese had no evidence to offer the Commission in support of the natural emergence theory. This was surprising because both the SARS-1 and MERS viruses had left copious traces in the environment. The intermediary host species of SARS-1 was identified within 4 months of the epidemic's outbreak, and the host of MERS within 9 months. Yet some 15 months after the SARS-2 pandemic began, and after a presumably intensive search, Chinese researchers had failed to find either the original bat population, or the intermediate species to which SARS-2 might have jumped, or any serological evidence that any Chinese population, including that of Wuhan, had ever been exposed to the virus prior to December 2019. Natural emergence remained a conjecture which, however plausible to begin with, had gained not a shred of supporting evidence in over a year. And as long as that remains the case, it's logical to pay serious attention to the alternative conjecture that SARS-2 escaped from a lab. Why would anyone want to create a novel virus capable of causing a pandemic? Ever since virologists gained the tools for manipulating a virus's genes, they have argued they could get ahead of a potential pandemic by exploring how close a given animal virus might be to making the jump to humans. And that justified lab experiments in enhancing the ability of dangerous animal viruses to infect people, virologists asserted. With this rationale, they have recreated the 1918 flu virus, shown how the almost extinct polio virus can be synthesized from its published DNA sequence, and introduced a smallpox gene into a related virus. These enhancements of viral capabilities are known blandly as gain-of-function experiments. With coronaviruses, there was particular interest in the spike proteins, which jut out all around the spherical surface of the virus and pretty much determine which species of animal it will target. In 2000, Dutch researchers, for instance, earned the gratitude of rodents everywhere by genetically engineering the spike protein of a mouse coronavirus so that it would attack only cats. Virologists started studying bat coronaviruses in earnest after these turned out to be the source of both the SARS-1 and MERS epidemics. In particular, researchers wanted to understand what changes needed to occur in a bat virus's spike proteins before it could infect people. Researchers at the Wuhan Institute of Virology, led by China's leading expert on bat viruses, Shizheng Li or Bat Lady, mounted frequent expeditions to the bat-infested caves of Yunnan in southern China and collected around 100 different bat coronaviruses. She then teamed up with Ralph S. Barrick, an eminent coronavirus researcher at the University of North Carolina. Their work focused on enhancing the ability of bat viruses to attack humans so as to examine the emergence potential, that is, the potential to infect humans, of circulating bat-CoVS coronaviruses. In pursuit of this aim, in November 2015 they created a novel virus by taking the backbone of the SARS-1 virus and replacing its spike protein with one from a bat virus, known as SHC014-CoV. This manufactured virus was able to infect the cells of the human airway, at least when tested against a lab culture of such cells. The SHC014-CoV/SARS-1 virus is known as a chimera because its genome contains genetic material from 2 strains of virus. If the SARS-2 virus were to have been cooked up in Xi's lab, then its direct prototype would have been the SHC014-CoV/SARS-1 chimera, the potential danger of which concerned many observers and prompted intense discussion. If the virus escaped, nobody could predict the trajectory, said Simon Wayne Hobson, a virologist at the Pasteur Institute in Paris. Barrick and she referred to the obvious risks in their paper but argued they should be weighed against the benefit of foreshadowing future spillovers. Scientific review panels, they wrote, may deem similar studies building chimeric viruses based on circulating strains too risky to pursue. Given various restrictions being placed on gain-of-function, GOF, research, matters had arrived in their view at a crossroads of GOF research concerns. The potential to prepare for and mitigate future outbreaks must be weighed against the risk of creating more dangerous pathogens. In developing policies moving forward, It is important to consider the value of the data generated by these studies and whether these types of chimeric virus studies warrant further investigation versus the inherent risks involved. That statement was made in 2015. From the hindsight of 2021, one can say that the value of gain-of-function studies in preventing the SARS-2 epidemic was zero. The risk was catastrophic, if indeed the SARS-2 virus was generated in a gain-of-function experiment. Inside the Wuhan Institute of Virology, Barack had developed, and taught Xi, a general method for engineering bat coronaviruses to attack other species. The specific targets were human cells grown in cultures and humanized mice. These laboratory mice, a cheap and ethical stand-in for human subjects, are genetically engineered to carry the human version of a protein called ACE2 that studs the surface of cells that line the airways. She returned to her lab at the Wuhan Institute of Virology and resumed the work she had started on genetically engineering coronaviruses to attack human cells. How can we be so sure? Because, by a strange twist in the story, her work was funded by the National Institute of Allergy and Infectious Diseases, NIAID, a part of the U.S. National Institutes of Health, NIH. And grant proposals that funded her work, which are a matter of public record, specify exactly what she planned to do with the money. The grants were assigned to the prime contractor, DASAC of the Eco Health Alliance, who subcontracted them to Xi. Here are extracts from the grants for fiscal years 2018 and 2019. CUF stands for coronavirus and S protein refers to the virus's spike protein. Test predictions of CUF interspecies transmission. Predictive models of host range, i.e. emergence potential, will be tested experimentally using reverse genetics, pseudovirus and receptor binding assays, and virus infection experiments across a range of cell cultures from different species and humanized mice. We will use S protein sequence data, infectious clone technology, in vitro and in vivo infection experiments and analysis of receptor binding to test the hypothesis that % divergence thresholds in S protein sequences predict spillover potential. What this means, in non-technical language, is that she set out to create novel coronaviruses with the highest possible infectivity for human cells. Her plan was to take genes that coded for spike proteins possessing a variety of measured affinities for human cells, ranging from high to low. She would insert these spike genes one by one into the backbone of a number of viral genomes, reverse genetics and infectious clone technology, creating a series of chimeric viruses. These chimeric viruses would then be tested for their ability to attack human cell cultures, in vitro, and humanized mice, in vivo. And this information would help predict the likelihood of spillover, the jump of a coronavirus from bats to people. The methodical approach was designed to find the best combination of coronavirus backbone and spike protein for infecting human cells. The approach could have generated SARS-2-like viruses, and indeed may have created the SARS-2 virus itself with the right combination of virus backbone and spike protein. It cannot yet be stated that she did or did not generate SARS-2 in her lab because her records have been sealed, but it seems she was certainly on the right track to have done so. It is clear that the Wuhan Institute of Virology was systematically constructing novel chimeric coronaviruses and was assessing their ability to infect human cells and human ACE2 expressing mice, says Richard H. Abright, a molecular biologist at Rutgers University and leading expert on biosafety. It is also clear, Abright said, that, depending on the constant genomic context chosen for analysis, this work could have produced SARS-CoV-2 or a proximal progenitor of SARS-CoV-2. Genomic context refers to the particular viral backbone used as the testbed for the spike protein. The lab escape scenario for the origin of the SARS-2 virus, as should by now be evident, is not mere hand-waving in the direction of the Wuhan Institute of Virology. It is a detailed proposal, based on the specific project being funded there by the NIAID. Even if the grant required the work plan described above, how can we be sure that the plan was in fact carried out? For that we can rely on the word of DASAC, who has been much protesting for the last 15 months that lab escape was a ludicrous conspiracy theory invented by China bashers. On December 9, 2019, before the outbreak of the pandemic became generally known, DASAC gave an interview in which he talked in glowing terms of how researchers at the Wuhan Institute of Virology had been reprogramming the spike protein and generating chimeric coronaviruses capable of infecting humanized mice. And we have now found, after 6 or 7 years of doing this, over 100 new SARS-related coronaviruses, very close to SARS, DASAC says around minute 28 of the interview. Some of them get into human cells in the lab, some of them can cause SARS disease in humanized mice models and are untreatable with therapeutic monoclonals, and you can't vaccinate against them with a vaccine. So, these are a clear and present danger. Interviewer: You say these are diverse coronaviruses and you can't vaccinate against them and no antivirals, so what do we do? Dasak, well, I think, coronaviruses you can manipulate them in the lab pretty easily. Spike protein drives a lot of what happened with coronavirus, in zoonotic risk. So you can get the sequence, you can build the protein, and we work a lot with Ralph Barrick at UNC to do this. Insert into the backbone of another virus and do some work in the lab. So you can get more predictive when you find a sequence. You've got this diversity. Now the logical progression for vaccines is, if you are going to develop a vaccine for SARS, people are going to use pandemic SARS, but let's insert some of these other things and get a better vaccine. The insertions he referred to perhaps included an element called the furin cleavage site, discussed below, which greatly increases viral infectivity for human cells. In disjointed style, Dasak is referring to the fact that once you have generated a novel coronavirus that can attack human cells, you can take the spike protein and make it the basis for a vaccine. One can only imagine Dasak's reaction when he heard of the outbreak of the epidemic in Wuhan a few days later. He would have known better than anyone the Wuhan Institute's goal of making bat coronaviruses infectious to humans, as well as the weaknesses in the Institute's defense against their own researchers becoming infected. But instead of providing public health authorities with the plentiful information at his disposal, he immediately launched a public relations campaign to persuade the world that the epidemic couldn't possibly have been caused by one of the Institute's souped-up viruses. The idea that this virus escaped from a lab is just pure baloney. It's simply not true, he declared in an April 2020 interview. The safety arrangements at the Wuhan Institute of Virology. Dasak was possibly unaware of, or perhaps he knew all too well, the long history of viruses escaping from even the best-run laboratories. The smallpox virus escaped 3 times from labs in England in the 1960s and 1970s, causing 80 cases and 3 deaths. Dangerous viruses have leaked out of labs almost every year since. Coming to more recent times, the SARS-1 virus has proved a true escape artist, leaking from laboratories in Singapore, Taiwan, and no less than 4 times from the Chinese National Institute of Virology in Beijing. One reason for SARS-1 being so hard to handle is that there were no vaccines available to protect laboratory workers. As Dasak mentioned in the December 19 interview quoted above, the Wuhan researchers too had been unable to develop vaccines against the coronaviruses they had designed to infect human cells. They would have been as defenseless against the SARS-2 virus, if it were generated in their lab, as their Beijing colleagues were against SARS-1. A second reason for the severe danger of novel coronaviruses has to do with the required levels of lab safety. There are 4 degrees of safety, designated BSL-1 to BSL-4, with BSL-4 being the most restrictive and designed for deadly pathogens like the Ebola virus. The Wuhan Institute of Virology had a new BSL-4 lab, but its state of readiness considerably alarmed the State Department inspectors who visited it from the Beijing embassy in 2018. The new lab has a serious shortage of appropriately trained technicians and investigators needed to safely operate this high-containment laboratory, the inspectors wrote in a cable of January 19, 2018. The real problem, however, was not the unsafe state of the Wuhan BSL-4 lab but the fact that virologists worldwide don't like working in BSL-4 conditions. You have to wear a space suit, do operations in closed cabinets, and accept that everything will take twice as long. So the rules assigning each kind of virus to a given safety level were laxer than some might think was prudent. Before 2020, the rules followed by virologists in China and elsewhere required that experiments with the SARS-1 and MERS viruses be conducted in BSL-3 conditions. But all other bat coronaviruses could be studied in BSL-2, the next level down. BSL-2 requires taking fairly minimal safety precautions, such as wearing lab coats and gloves, not sucking up liquids in a pipette, and putting up biohazard warning signs. Yet again, a function experiment conducted in BSL-2 might produce an agent more infectious than either SARS-1 or MERS. And if it did, then lab workers would stand a high chance of infection, especially if unvaccinated. Much of Xi's work on gain of function in coronaviruses was performed at the BSL-2 safety level, as is stated in her publications and other documents. She has said in an interview with Science magazine that, TE coronavirus research in our laboratory is conducted in BSL-2 or BSL-3 laboratories. It is clear that some or all of this work was being performed using a biosafety standard biosafety level 2, the biosafety level of a standard U.S. dentist's office that would pose an unacceptably high risk of infection of laboratory staff upon contact with a virus having the transmission properties of SARS-CoV-2, Bright says. It also is clear, he adds, that this work never should have been funded and never should have been performed. This is a view he holds regardless of whether or not the SARS-2 virus ever saw the inside of a lab. Concern about safety conditions at the Wuhan lab was not, it seems, misplaced. According to a fact sheet issued by the State Department on January 15, 2021, the U.S. government has reason to believe that several researchers inside the WIV became sick in autumn 2019, before the first identified case of the outbreak, with symptoms consistent with both COVID-19 and common seasonal illnesses. David Asher, a fellow of the Hudson Institute and former consultant to the State Department, provided more detail about the incident at a seminar. Knowledge of the incident came from a mix of public information and some high-end information collected by our intelligence community, he said. 3 people working at a BSL-3 lab at the Institute fell sick within a week of each other with severe symptoms that required hospitalization. This was the first known cluster that we're aware of victims of what we believe to be COVID-19. Influenza could not completely be ruled out but seemed unlikely in the circumstances, he said. Comparing the rival scenarios of SARS-2 origin. The evidence above adds up to a serious case that the SARS-2 virus could have been created in a lab, from which it then escaped. But the case, however substantial, falls short of proof. Proof would consist of evidence from the Wuhan Institute of Virology, or related labs in Wuhan, that SARS-2 or a predecessor virus was under development there. For lack of access to such records, another approach is to take certain salient facts about the SARS-2 virus and ask how well each is explained by the 2 rival scenarios of origin, those of natural emergence and lab escape. Here are 4 tests of the 2 hypotheses. A couple have some technical detail, but these are among the most persuasive for those who may care to follow the argument. 1. The place of origin. Start with geography. The 2 closest known relatives of the SARS-2 virus were collected from bats living in caves in Yunnan, a province of southern China. If the SARS-2 virus had first infected people living around the Yunnan caves, that would strongly support the idea that the virus had spilled over to people naturally. But this isn't what happened. The pandemic broke out 1,500 kilometers away, in Wuhan. Beta coronaviruses, the family of bat viruses to which SARS-2 belongs, infect the horseshoe bat Rhinolepis affinis, which ranges across southern China. The bat's range is 50 kilometers, so it's unlikely that any made it to Wuhan. In any case, the first cases of the COVID-19 pandemic probably occurred in September, when temperatures in Hubei province are already cold enough to send bats into hibernation. What if the bat viruses infected some intermediate host first? You would need a long-standing population of bats in frequent proximity with an intermediate host, which in turn must often cross paths with people. All these exchanges of virus must take place somewhere outside Wuhan, a busy metropolis which so far as is known is not a natural habitat of Rhinolepis bat colonies. The infected person, or animal, carrying this highly transmissible virus must have traveled to Wuhan without infecting anyone else. No one in his or her family got sick. If the person jumped on a train to Wuhan, no fellow passengers fell ill. It's a stretch, in other words, to get the pandemic to break out naturally outside Wuhan and then, without leaving any trace, to make its first appearance there. For the lab escape scenario, a Wuhan origin for the virus is a no-brainer. Wuhan is home to China's leading center of coronavirus research where, as noted above, researchers were genetically engineering bat coronaviruses to attack human cells. They were doing so under the minimal safety conditions of a BSL-2 lab. If a virus with the unexpected infectiousness of SARS-2 had been generated there, its escape would be no surprise. 2. Natural history and evolution. The initial location of the pandemic is a small part of a larger problem, that of its natural history. Viruses don't just make one-time jumps from one species to another. The coronavirus spike protein, adapted to attack bat cells, needs repeated jumps to another species, most of which fail, before it gains a lucky mutation. Mutation: a change in one of its RNA units causes a different amino acid unit to be incorporated into its spike protein and makes the spike protein better able to attack the cells of some other species. Through several more such mutation-driven adjustments, the virus adapts to its new host, say some animal with which bats are in frequent contact. The whole process then resumes as the virus moves from this intermediate host to people. In the case of SARS-1, researchers have documented the successive changes in its spike protein as the virus evolved step by step into a dangerous pathogen. After it had gotten from bats into civets, there were 6 further changes in its spike protein before it became a mild pathogen in people. After a further 14 changes, the virus was much better adapted to humans, and with a further 4, the epidemic took off. But when you look for the fingerprints of a similar transition in SARS-2, a strange surprise awaits. The virus has changed hardly at all, at least until recently. From its very first appearance, it was well adapted to human cells. Researchers led by Alina Chan of the Broad Institute compared SARS-2 with late-stage SARS-1, which by then was well adapted to human cells, and found that the two viruses were similarly well adapted. By the time SARS-CoV-2 was first detected in late 2019, it was already pre-adapted to human transmission to an extent similar to late epidemic SARS-CoV, they wrote. Even those who think lab origin unlikely agree that SARS-2 genomes are remarkably uniform. Barrick writes that early strains identified in Wuhan, China, showed limited genetic diversity, which suggests that the virus may have been introduced from a single source. A single source would of course be compatible with lab escape, less so with the massive variation in selection which is evolution's hallmark way of doing business. The uniform structure of SARS-2 genomes gives no hint of any passage through an intermediate animal host, and no such host has been identified in nature. Proponents of natural emergence suggest that SARS-2 incubated in a yet-to-be-found human population before gaining its special properties. Or that it jumped to a host animal outside China. All these conjectures are possible, but strained. Proponents of a lab leak have a simpler explanation. SARS-2 was adapted to human cells from the start because it was grown in humanized mice or in lab cultures of human cells, just as described in Daszak's grant proposal. Its genome shows little diversity because the hallmark of lab cultures is uniformity. Proponents of laboratory escape joke that of course the SARS-2 virus infected an intermediary host species before spreading to people, and that they have identified it a humanized mouse from the Wuhan Institute of Virology. 3. The furin cleavage site. The furin cleavage site is a minute part of the virus's anatomy but one that exerts great influence on its infectivity. It sits in the middle of the SARS-2 spike protein. It also lies at the heart of the puzzle of where the virus came from. The spike protein has two subunits with different roles. The first, called S1, recognizes the virus's target, a protein called angiotensin-converting enzyme 2, or ACE2, which studs the surface of cells lining the human airways. The second, S2, helps the virus, once anchored to the cell, to fuse with the cell's membrane. After the virus's outer membrane has coalesced with that of the stricken cell, the viral genome is injected into the cell, hijacks its protein-making machinery and forces it to generate new viruses. But this invasion cannot begin until the S1 and S2 subunits have been cut apart. And there, right at the S1/S2 junction, is the furin cleavage site that ensures the spike protein will be cleaved in exactly the right place. The virus, a model of economic design, does not carry its own cleaver. It relies on the cell to do the cleaving for it. Human cells have a protein cutting tool on their surface known as furin. Furin will cut any protein chain that carries its signature target cutting site. This is the sequence of amino acid units proline, arginine, arginine, alanine, or PRRA in the code that refers to each amino acid by a letter of the alphabet. PRRA is the amino acid sequence at the core of SARS-2's furin cleavage site. Viruses have all kinds of clever tricks, so why does the furin cleavage site stand out? Because of all known SARS-related beta coronaviruses, only SARS-2 possesses a furin cleavage site. All the other viruses have their S2 unit cleaved at a different site and by a different mechanism. How then did SARS-2 acquire its furin cleavage site? Either the site evolved naturally, or it was inserted by researchers at the S1/S2 junction in a gain-of-function experiment. Consider natural origin first. Two ways viruses evolve are by mutation and by recombination. Mutation is the process of random change in DNA, or RNA for coronaviruses, that usually results in one amino acid in a protein chain being switched for another. Many of these changes harm the virus, but natural selection retains the few that do something useful. Mutation is the process by which the SARS-1 spike protein gradually switched its preferred target cells from those of bats to civets, and then to humans. Mutation seems a less likely way for SARS-2's furin cleavage site to be generated, even though it can't completely be ruled out. The site's 4 amino acid units are all together, and all at just the right place in the S1/S2 junction. Mutation is a random process triggered by copying errors, when new viral genomes are being generated, or by chemical decay of genomic units. So it typically affects single amino acids at different spots in a protein chain. A string of amino acids like that of the furin cleavage site is much more likely to be acquired altogether through a quite different process known as recombination. Recombination is an inadvertent swapping of genomic material that occurs when two viruses happen to invade the same cell, and their progeny are assembled with bits and pieces of RNA belonging to the other. Beta coronaviruses will only combine with other beta coronaviruses but can acquire, by recombination, almost any genetic element present in the collective genomic pool. What they cannot acquire is an element the pool does not possess. And no known SARS-related beta coronavirus, the class to which SARS-2 belongs, possesses a furin cleavage site. Proponents of natural emergence say SARS-2 could have picked up the site from some as yet unknown beta coronavirus. But bat SARS-related beta coronaviruses evidently don't need a furin cleavage site to infect bat cells, so there's no great likelihood that any in fact possesses one, and indeed none has been found so far. The proponent's next argument is that SARS-2 acquired its furin cleavage site from people. A predecessor of SARS-2 could have been circulating in the human population for months or years until at some point it acquired a furin cleavage site from human cells. It would then have been ready to break out as a pandemic. If this is what happened, there should be traces in hospital surveillance records of the people infected by the slowly evolving virus. But none has so far come to light. According to the WHO report on the origins of the virus, the Sentinel hospitals in Hubei province, home of Wuhan, routinely monitor influenza-like illnesses, and no evidence to suggest substantial SARS-CoV-2 transmission in the months preceding the outbreak in December was observed. So it's hard to explain how the SARS-2 virus picked up its furin cleavage site naturally, whether by mutation or recombination. That leaves a gain-of-function experiment. For those who think SARS-2 may have escaped from a lab, explaining the furin cleavage site is no problem at all. Since 1992, the virology community has known that the one sure way to make a virus deadlier is to give it a furin cleavage site at the S1/S2 junction in the laboratory, writes Stephen Key, a biotech entrepreneur interested in the origins of SARS-2. At least 11 gain-of-function experiments, adding a furin site to make a virus more infective, are published in the open literature, including, by Dr. Zhengli Shi, head of coronavirus research at the Wuhan Institute of Virology. 4. A question of codons. There's another aspect of the furin cleavage site that narrows the path for a natural emergence origin even further. As everyone knows, or may at least recall from high school, the genetic code uses 3 units of DNA to specify each amino acid unit of a protein chain. When read in groups of 3, the 4 different kinds of DNA unit can specify 4x4x4 or 64 different triplets, or codons as they are called. Since there are only 20 kinds of amino acid, there are more than enough codons to go around, allowing some amino acids to be specified by more than one codon. The amino acid arginine, for instance, can be designated by any of the 6 codons CGU, CGC, CGA, CGG, AGO or AGG, where A, U, G and C stand for the 4 different kinds of unit in RNA. Here's where it gets interesting. Different organisms have different codon preferences. Human cells like to designate arginine with the codon CGT, CGC or CGG. But CGG is coronavirus's least popular codon for arginine. Keep that in mind when looking at how the amino acids in the furin cleavage site are encoded in the SARS-2 genome. Now the functional reason why SARS-2 has a furin cleavage site, and its cousin viruses don't, can be seen by lining up, in a computer, the string of nearly 30,000 nucleotides in its genome with those of its cousin coronaviruses, of which the closest so far known is one called RAC13. Compared with RAC13, SARS-2 has a 12 nucleotide insert right at the S1/S2 junction. The insert is the sequence TCCT-CGG-CGGGC. The CCT codes for proline, the 2 CGGs for 2 arginines, and the GC is the beginning of a GCA codon that codes for alanine. There are several curious features about this insert, but the oddest is that of the 2 side-by-side CGG codons. Only 5% of SARS-2's arginine codons are CGG, and the double codon CGG-CGG has not been found in any other beta coronavirus. So how did SARS-2 acquire a pair of arginine codons that are favored by human cells but not by coronaviruses? Proponents of natural emergence have an uphill task to explain all the features of SARS-2's furin cleavage site. They have to postulate a recombination event at a site on the virus's genome where recombinations are rare, and the insertion of a 12 nucleotide sequence with a double arginine codon unknown in the beta coronavirus repertoire, at the only site in the genome that would significantly expand the virus's infectivity. Yes, but your wording makes this sound unlikely. Viruses are specialists at unusual events, is the riposte of David L. Robertson, a virologist at the University of Glasgow who regards lab escape as a conspiracy theory. Recombination is naturally very frequent in these viruses. There are recombination breakpoints in the spike protein, and these codons appear unusual exactly because we've not sampled enough. Robertson is correct that evolution is always producing results that may seem unlikely but in fact are not. Viruses can generate untold numbers of variants, but we see only the 1 in a billion that natural selection picks for survival. But this argument could be pushed too far. For instance, any result of a gain-of-function experiment could be explained as one that evolution would have arrived at in time. And the numbers game can be played the other way. For the furin cleavage site to arise naturally in SARS-2, a chain of events has to happen, each of which is quite unlikely for the reasons given above. A long chain with several improbable steps is unlikely to ever be completed. For the lab escape scenario, the double CGG codon is no surprise. The human preferred codon is routinely used in labs. So anyone who wanted to insert a furin cleavage site into the virus's genome would synthesize the prRNA-making sequence in the lab and would be likely to use CGG codons to do so. One problem with this idea, though, is that if SARS-2 jumped from bats to people in a single leap and hasn't changed much since, it should still be good at infecting bats. And it seems it isn't. Tested bat species are poorly infected by SARS-CoV-2 and they are therefore unlikely to be the direct source for human infection, write a scientific group skeptical of natural emergence. Still, Robertson may be onto something. The bat coronaviruses of the Yunnan caves can infect people directly. In April 2012, 6 miners clearing bat guano from the Mojiang mine contracted severe pneumonia with COVID-19-like symptoms and 3 eventually died. A virus isolated from the Mojiang mine, called Rat13, is still the closest known relative of SARS-2. Much mystery surrounds the origin, reporting and strangely low affinity of Rat13 for bat cells, as well as the nature of 8 similar viruses that she reports she collected at the same time but has not yet published despite their great relevance to the ancestry of SARS-2. But all that is a story for another time. The point here is that bat viruses can infect people directly, though only in special conditions. So who else, besides miners excavating bat guano, comes into particularly close contact with bat coronaviruses? Well, coronavirus researchers do. She says she and her group collected more than 1,300 bat samples during some 8 visits to the Mojiang cave between 2012 and 2015, and there were doubtless many expeditions to other Yunnan caves. Imagine the researchers making frequent trips from Wuhan to Yunnan and back, stirring up bat guano in dark caves and mines, and now you begin to see a possible missing link between the two places. Researchers could have gotten infected during their collecting trips, or while working with the new viruses at the Wuhan Institute of Virology. The virus that escaped from the lab would have been a natural virus, not one cooked up by gain of function. The direct from bats thesis is a chimera between the natural emergence and lab escape scenarios. It's a possibility that can't be dismissed. But against it are the facts that 1, both SARS-2 and Rat13 seem to have only feeble affinity for bat cells, so one can't be fully confident that either ever saw the inside of a bat, and 2, the theory is no better than the natural emergence scenario at explaining how SARS-2 gained its furin cleavage site, or why the furin cleavage site is determined by human-preferred arginine codons instead of by the bat-preferred codons. Where we are so far, neither the natural emergence nor the lab escape hypothesis can yet be ruled out. There is still no direct evidence for either. So no definitive conclusion can be reached. That said, the available evidence leans more strongly in one direction than the other. Readers will form their own opinion. But it seems to me that proponents of lab escape can explain all the available facts about SARS-2 considerably more easily than can those who favor natural emergence. It's documented that researchers at the Wuhan Institute of Virology were doing gain-of-function experiments designed to make coronaviruses infect human cells and humanized mice. This is exactly the kind of experiment from which a SARS-2-like virus could have emerged. The researchers were not vaccinated against the viruses under study, and they were working in the minimal safety conditions of a BSL-2 laboratory. So escape of a virus would not be at all surprising. In all of China, the pandemic broke out on the doorstep of the Wuhan Institute. The virus was already well adapted to humans, as expected for a virus grown in humanized mice. It possessed an unusual enhancement, a furin cleavage site, which is not possessed by any other known SARS-related beta coronavirus, and this site included a double arginine codon also unknown among beta coronaviruses. What more evidence could you want, aside from the presently unobtainable lab records documenting SARS-2's creation? Proponents of natural emergence have a rather harder story to tell. The plausibility of their case rests on a single surmise, the expected parallel between the emergence of SARS-2 and that of SARS-1 and MERS. But none of the evidence expected in support of such a parallel history has yet emerged. No one has found the bat population that was the source of SARS-2, if indeed it ever infected bats. No intermediate host has presented itself, despite an intensive search by Chinese authorities that included the testing of 80,000 animals. There is no evidence of the virus making multiple independent jumps from its intermediate host to people, as both the SARS-1 and MERS viruses did. There is no evidence from hospital surveillance records of the epidemic gathering strength in the population as the virus evolved. There is no explanation of why a natural epidemic should break out in Wuhan and nowhere else. There is no good explanation of how the virus acquired its furin cleavage site, which no other SARS-related beta coronavirus possesses, nor why the site is composed of human-preferred codons. The natural emergence theory battles a bristling array of implausibilities. The records of the Wuhan Institute of Virology certainly hold much relevant information. But Chinese authorities seem unlikely to release them given the substantial chance that they incriminate the regime in the creation of the pandemic. Absent the efforts of some courageous Chinese whistleblower, we may already have at hand just about all of the relevant information we are likely to get for a while. So it's worth trying to assess responsibility for the pandemic, at least in a provisional way, because the paramount goal remains to prevent another one. Even those who aren't persuaded that lab escape is the more likely origin of the SARS-2 virus may see reason for concern about the present state of regulation governing gain-of-function research. There are two obvious levels of responsibility: the first, for allowing virologists to perform gain-of-function experiments, offering minimal gain and vast risk; the second, if indeed SARS-2 was generated in a lab, for allowing the virus to escape and unleash a worldwide pandemic. Here are the players who seem most likely to deserve blame. I have been using the Wuhan Institute of Virology as a shorthand for all virological activities in Wuhan. It's possible that SARS-2 was generated in some other Wuhan lab, perhaps in an attempt to make a vaccine that worked against all coronaviruses. But until the role of other Chinese virologists is clarified, she is the public face of Chinese work on coronaviruses, and provisionally she and her colleagues will stand first in line for opprobrium. 2. Chinese authorities. China's central authorities did not generate SARS-2, but they sure did their utmost to conceal the nature of the tragedy and China's responsibility for it. They suppressed all records at the Wuhan Institute of Virology and closed down its virus databases. They released a trickle of information, much of which may have been outright false or designed to misdirect and mislead. They did their best to manipulate the WHO's inquiry into the virus's origins, and led the Commission's members on a fruitless runaround. So far they have proved far more interested in deflecting blame than in taking the steps necessary to prevent a second pandemic. 3. The worldwide community of virologists. Virologists around the world are a loose-knit professional community. They write articles in the same journals. They attend the same conferences. They have common interests in seeking funds from governments and in not being overburdened with safety regulations. Virologists knew better than anyone the dangers of gain-of-function research. But the power to create new viruses, and the research funding obtainable by doing so, was too tempting. They pushed ahead with gain-of-function experiments. They lobbied against the moratorium imposed on federal funding for gain-of-function research in 2014, and it was raised in 2017. The benefits of the research in preventing future epidemics have so far been nil, the risks vast. If research on the SARS-1 and MERS viruses could only be done at the BSL-3 safety level, it was surely illogical to allow any work with novel coronaviruses at the lesser level of BSL-2. Whether or not SARS-2 escaped from a lab, virologists around the world have been playing with fire. Their behavior has long alarmed other biologists. In 2014, scientists calling themselves the Cambridge Working Group urged caution on creating new viruses. In prescient words, they specified the risk of creating a SARS-2-like virus. Accident risks with newly created potential pandemic pathogens raise grave new concerns, they wrote. Laboratory creation of highly transmissible, novel strains of dangerous viruses, especially but not limited to influenza, poses substantially increased risks. An accidental infection in such a setting could trigger outbreaks that would be difficult or impossible to control. When molecular biologists discovered a technique for moving genes from one organism to another, they held a public conference at Asilomar in 1975 to discuss the possible risks. Despite much internal opposition, they drew up a list of stringent safety measures that could be relaxed in future and duly were when the possible hazards had been better assessed. When the CRISPR technique for editing genes was invented, biologists convened a joint report by the US, UK and Chinese National Academies of Science to urge restraint on making heritable changes to the human genome. Biologists who invented gene drives have also been open about the dangers of their work and have sought to involve the public. You might think the SARS-2 pandemic would spur virologists to re-evaluate the benefits of gain-of-function research, even to engage the public in their deliberations. But no. Many virologists deride lab escape as a conspiracy theory, and others say nothing. They have barricaded themselves behind a Chinese wall of silence which so far is working well to allay, or at least postpone, journalists' curiosity and the public's wrath. Professions that cannot regulate themselves deserve to get regulated by others, and this would seem to be the future that virologists are choosing for themselves. 4. The U.S. role in funding the Wuhan Institute of Virology. From June 2014 to May 2019, DASAC's Eco-Health Alliance had a grant from the National Institute of Allergy and Infectious Diseases, NIAID, part of the National Institutes of Health, to do gain-of-function research with coronaviruses at the Wuhan Institute of Virology. Whether or not SARS-2 is the product of that research, it seems a questionable policy to farm out high-risk research to unsafe foreign labs using minimal safety precautions. And if the SARS-2 virus did indeed escape from the Wuhan Institute, then the NIH will find itself in the terrible position of having funded a disastrous experiment that led to death of more than 3 million worldwide, including more than 500,000 of its own citizens. The responsibility of the NIAID and NIH is even more acute because for the first 3 years of the grant to Eco-Health Alliance, there was a moratorium on funding gain-of-function research. Why didn't the two agencies therefore halt the federal funding, as apparently required to do so by law? Because someone wrote a loophole into the moratorium. The moratorium specifically barred funding any gain-of-function research that increased the pathogenicity of the flu, MERS, or SARS viruses. But then a footnote on page 2 of the moratorium document states that, an exception from the research pause may be obtained if the head of the USG funding agency determines that the research is urgently necessary to protect the public health or national security. This seems to mean that either the director of the NIAID, Anthony Fauci, or the director of the NIH, Francis Collins, or maybe both, would have invoked the footnote in order to keep the money flowing to Xi's gain-of-function research. Unfortunately, the NIAID director and the NIH director exploited this loophole to issue exemptions to projects subject to the pause, preposterously asserting the exempted research was urgently necessary to protect public health or national security, thereby nullifying the pause, Ebright said in an interview with Independent Science News. When the moratorium was ended in 2017, it didn't just vanish but was replaced by a reporting system, the Potential Pandemic Pathogens Control and Oversight (PCO) framework, which required agencies to report for review any dangerous gain-of-function work they wished to fund. According to Ebright, both Collins and Fauci have declined to flag and forward proposals for risk-benefit review, thereby nullifying the PACO framework. In his view, the two officials, in dealing with the moratorium and the ensuing reporting system, have systematically thwarted efforts by the White House, the Congress, scientists, and science policy specialists to regulate GOF, gain-of-function, research of concern. Possibly the two officials had to take into account matters not evident in the public record, such as issues of national security. Perhaps funding the Wuhan Institute of Virology, which is believed to have ties with Chinese military virologists, provided a window into Chinese biowarfare research. But whatever other considerations may have been involved, the bottom line is that the National Institutes of Health was supporting gain-of-function research, of a kind that could have generated the SARS-2 virus, in an unsupervised foreign lab that was doing work in BSL-2 biosafety conditions. The prudence of this decision can be questioned, whether or not SARS-2 and the death of 3 million people were the result of it, which emphasizes the need for some better system of control. In conclusion, if the case that SARS-2 originated in a lab is so substantial, why isn't this more widely known? As may now be obvious, there are many people who have reason not to talk about it. The list is led, of course, by the Chinese authorities. But virologists in the United States and Europe have no great interest in igniting a public debate about the gain-of-function experiments that their community has been pursuing for years. Nor have other scientists stepped forward to raise the issue. Government research funds are distributed on the advice of committees of scientific experts drawn from universities. Anyone who rocks the boat by raising awkward political issues runs the risk that their grant will not be renewed and that a research career will be ended. Maybe good behavior is rewarded with the many perks that slosh around the distribution system. And if you thought that Anderson and Daszak might have blotted their reputation for scientific objectivity after their partisan attacks on the lab escape scenario, look at the second and third names on this list of recipients of an $82 million grant announced by the National Institute of Allergy and Infectious Diseases in August 2020. The U.S. government shares a strange common interest with the Chinese authorities. Neither is keen on drawing attention to the fact that Xi's coronavirus work was funded by the U.S. National Institutes of Health. One can imagine the behind-the-scenes conversation in which the Chinese government says, "If this research was so dangerous, why did you fund it, and on our territory too?" To which the U.S. side might reply, "Looks like it was you who let it escape." But do we really need to have this discussion in public? Fauci is a longtime public servant who served with integrity under President Trump and has resumed leadership in the Biden administration in handling the COVID-19 epidemic. Congress, no doubt understandably, may have little appetite for hauling him over the coals for the apparent lapse of judgment in funding gain-of-function research in Wuhan. To these serried walls of silence must be added that of the mainstream media. To my knowledge, no major newspaper or television network has yet provided readers with an in-depth news story of the lab escape scenario, such as the one you have just read, although some have run brief editorials or opinion pieces. One might think that any plausible origin of a virus that has killed 3 million people would merit a serious investigation. Or that the wisdom of continuing gain-of-function research, regardless of the virus's origin, would be worth some probing. Or that the funding of gain-of-function research by the NIH and NIAID during a moratorium on such funding would bear investigation. What accounts for the media's apparent lack of curiosity? The virologist's omerta is one reason. Science reporters, unlike political reporters, have little innate skepticism of their sources' motives. Most see their role largely as purveying the wisdom of scientists to the unwashed masses. So when their sources won't help, these journalists are at a loss. Another reason, perhaps, is the migration of much of the media toward the left of the political spectrum. Because President Trump said the virus had escaped from a Wuhan lab, editors gave the idea little credence. They joined the virologists in regarding lab escape as a dismissible conspiracy theory. During the Trump administration, they had no trouble in rejecting the position of the intelligence services that lab escape could not be ruled out. But when Avril Haines, President Biden's Director of National Intelligence, said the same thing, she too was largely ignored. This is not to argue that editors should have endorsed the lab escape scenario, merely that they should have explored the possibility fully and fairly. People around the world who have been pretty much confined to their homes for the last year might like a better answer than their media are giving them. Perhaps one will emerge in time. After all, the more months pass without the natural emergence theory gaining a shred of supporting evidence, the less plausible it may seem. Perhaps the international community of virologists will come to be seen as a false and self-interested guide. The common-sense perception that a pandemic breaking out in Wuhan might have something to do with a Wuhan lab cooking up novel viruses of maximal danger in unsafe conditions could eventually displace the ideological insistence that whatever Trump said can't be true. And then let the reckoning begin. Notes. 1. This quotation was added to the article after initial publication. Acknowledgements. The first person to take a serious look at the origins of the SARS-2 virus was Yuri Diagin, a biotech entrepreneur in Russia and Canada. In a long and brilliant essay, he dissected the molecular biology of the SARS-2 virus and raised, without endorsing, the possibility that it had been manipulated. The essay, published on April 22, 2020, provided a roadmap for anyone seeking to understand the virus's origins. Diagin packed so much information and analysis into his essay that some have doubted it could be the work of a single individual and suggested some intelligence agency must have authored it. But the essay is written with greater lightness and humor than I suspect are ever found in CIA or KGB reports, and I see no reason to doubt that Diagin is its very capable sole author. In Diagin's wake have followed several other skeptics of a virologist's orthodoxy. Nikolai Petrovsky calculated how tightly the SARS-2 virus binds to the ACE2 receptors of various species and found to his surprise that it seemed optimized for the human receptor, leading him to infer the virus might have been generated in a laboratory. Alina Chan published a paper showing that SARS-2 from its first appearance was very well adapted to human cells. One of the very few establishment scientists to have questioned the virologist's absolute rejection of lab escape is Richard A. Bright, who has long warned against the dangers of gain-of-function research. Another is David A. Relman of Stanford University. Even though strong opinions abound, none of these scenarios can be confidently ruled in or ruled out with currently available facts, he wrote. Kudos to Robert Redfield, former director of the Centers for Disease Control and Prevention, who told CNN on March 26, 2021 that the most likely cause of the epidemic was from a laboratory, because he doubted that a bat virus could become an extreme human pathogen overnight, without taking time to evolve, as seemed to be the case with SARS-2. Stephen Key, a physician researcher, has applied statistical and bioinformatic tools to ingenious explorations of the virus's origin, showing for instance how the hospitals receiving the early patients are clustered along the Wuhan No. 2 subway line, which connects the Institute of Virology at one end with the International Airport at the other, the perfect conveyor belt for distributing the virus from lab to globe. In June 2020, Melton Leitenberg published an early survey of the evidence favoring lab escape from gain-of-function research at the Wuhan Institute of Virology. Many others have contributed significant pieces of the puzzle. Truth is the daughter, Said Francis Bacon, not of authority but time. The efforts of people such as those named above are what makes it so.